Okay, so welcome to the animal lecture. This one is going to be a little bit longer than some of our other lectures. There's a lot of information happening in this chapter, so definitely make sure that you are reading through the chapter, but hopefully this takes some of that information and kind of explains it a little bit better as well, kind of putting things in perspective. So this is going to be a long one, so hopefully all of this information makes sense. If it doesn't, please feel free to let me know. Happy to try and explain it a little bit differently. So first of all, we're going to talk about what an animal is. Then we'll kind of go into animal evolution and then go through the diversity of animals that we see. So animals are eukaryotic, so there are no prokaryotic animals. They all have those membrane-bound organelles. They all have that linear DNA that's inside that nucleus, right? So we are definitely fully in the eukaryotic domain here, no prokaryotes. Animals are also multicellular. There's no such thing as a unicellular animal. So all animals are multicellular. So this picture down here is just showing a developing embryo. You can see that there are multiple cells there. So all animals are multicellular. Animals also don't have cell walls. So we saw cell walls in our plant group and some of our other groups. But even though animals don't have cell walls, they still have some sort of structural support. So it's a variety of proteins that animals use for structural support. The main one that you see in animals is collagen. Collagen is only found in animals. It's not found in any other kingdoms. So that's one of the ways that animals keep that structural support without those cell walls. And then animals are also heterotrophic. So this is like the fungi that we saw. Fungi are also heterotrophic and can't make their own food. So they have to eat other organic matter to get those nutrients. So within animals, they are all heterotrophic, but there are lots of strategies for being heterotrophic. So you can be herbivorous, you can be an herbivore like this deer over here. You can be a carnivore like this lion. This badger here is representing our omnivores. So we are also omnivores. Most of us are, but some of us are more herbivorous or carnivorous. But omnivores eat both plant and animal material. And then we can also have detritivores, like this earthworm here. So eating decaying organic matter or dead organic matter. Unlike fungi, however, animals are going to digest their food internally. So with fungi, it was external digestion, meaning that they would put enzymes out of the cell into the environment to digest the food and then obtain the nutrients. Animals are going to ingest the food and then digest the food. So it was similar to fungi in the sense that we're heterotrophic, different than fungi in the sense that we are digesting internally rather than externally. Okay, so animals evolved from protists, from single-celled protists. These protists were called, or are called choanoflagellates. This is really obvious when we look at sponges, which are the most basal animals. So you can see that this is a representation of this protist that animals evolved from. And then this right here is a type of cell that we find in sponges, inside the walls of sponges. So you can see that these are very, very similar. So that's where we see those similarities in those sponges, those most primitive animals. And if we look at this on a phylogenetic tree, we can see that we have our eukaryotes here. We have this split into choanoflagellates, and we have a split into animals. So choanoflagellates are the most close protist ancestor we have to animals. And then within animals, we break off into sponges and then into other animals. So sponges are the most basal animals that we have, and those are the most similar to these protists, these choanoflagellates. So the entire body of the sponge doesn't necessarily look like these choanoflagellates, but there are special types of cells within the sponge that we'll talk about in the sponge section that look very similar to these protists. So we're going to walk through the animal phylogenetic tree and kind of look at the major splits that occurred in animal evolution. And then we're going to go through all of these evolutionary events and kind of talk about what those traits look like. So within our animals, so we're not talking protists here, these are just our animals. The first big split that we see is the evolution of true tissues and the evolution of symmetry. So animals that don't have any true tissues and also don't have any symmetry, that is in the group Parazoa, and those are our sponges, phylum Porifera. So all of our phyla are going to line up over here on the right side of the screen. So we get this split into no tissues versus yes, we have tissues. So that's going to be up here. And then all of these animals that have tissues are also symmetric in some way. So within our true tissue animals and our symmetric animals, we have a split into different types of symmetry and the evolution of different types of germ layers. So down here, we have radially symmetric organisms. Those are going to be phylum Cnidaria, things like jellyfish. And then everything else is bilaterally symmetrical. So you also see these words up here, triploblastic and diploblastic. That's referring to these germ layers. So the ways that tissues develop during embryogenesis when that embryo is developing. Our radially symmetrical organisms do things a little bit differently than our bilaterally symmetrical organisms. So we'll talk about these in a couple slides, but just know that when it comes to tissue development in embryos, bilaterally symmetrical organisms are different than radially symmetrical organisms. And then within our bilaterally symmetrical organisms, our triploblastic organisms, we have two different types of embryonic development. And again, we're going to talk about this in a couple slides, but there are two different types of embryonic development. And depending on how you develop, you are going to be classified as either a protostome or a deuterostome. So those are the different types of embryonic development there. Within our protostomes, we have two further classifications. And this is just done by DNA evidence, and we're going to talk about these in a few slides as well. Just know that they do split for now, and we'll talk about what those mean in a little bit. But those are Ecdysozoa and Lophotrochozoa. Those are our two splits that we have within our protostomes. So the two phyla we're going to be talking about within our Ecdysozoan group are arthropods and nematodes. Within our Lophotrochozoa group, we've got a couple phylum. We're today going to talk about mollusks, annelids, and these flatworms down here. And then within the deuterostome development, again, this is a type of embryonic development. We have phylum Echinodermata, so these are things like sea stars, so starfish there in Echinodermata. And then phylum Chordata, which includes the vertebrates like us. So we're down here, we have a deuterostome development and are bilaterally symmetrical. So we'll talk about the chordates last. We'll go through these other phyla first. So this is the big picture roadmap for where we're going today. We're going to talk about these phyla. So true tissues. So we talked about how most of our animals have these true tissues. That means that there are specialized cells that have grouped together to perform some sort of specialized function. So the phylum Porifera, the sponges, don't have these specialized cells that perform these specialized functions. These are things like nervous tissue, muscle tissue, connective tissue, epithelial tissue. So all of those types of tissues are in our groups of animals that have tissues. Sponges do not have these specialized cell groups. Symmetry is referring to being made up of similar parts that face each other around an axis. So our sponges are considered to be asymmetrical. Asymmetrical meaning there is no symmetry. So that's like this sponge down here. There's no symmetry happening in that sponge. The other option for symmetry is radial symmetry. So we only saw one phyla with radial symmetry. That's phylum Cnidaria, so things like our jellyfish. So that is going to be like this, where they are symmetrical around a central axis. You can kind of put multiple planes of symmetry within this organism, and you have identical halves. And then bilateral symmetry, so that's the one that we have, and most animals that we're going to talk about today have this bilateral symmetry. You have a right side and a left side, right? You can put a plane of symmetry down the middle of this organism here, and you're going to have an identical right side and left side. That also allows us to sort of give some other descriptors of, sort of general parts of the body. So anterior versus posterior. So anterior referring to this head end, posterior referring to the tail end. And then we can also classify things as dorsal versus ventral. Dorsal referring to the back side of the organism, ventral referring to the tummy side of the organism. But we just have this one plane of symmetry that we can put through the animal that gives us a right side and a left side. So three types of symmetry, two types of symmetry, and then an option to not be symmetrical. So within our symmetrical organisms, so this is just talking about our radially symmetrical and our bilaterally symmetrical organisms, we have layers of tissue that develop during embryonic development that have different number of layers. So the tissues begin to layer when the embryo is developing, and these layers are called germ layers. These layers will eventually develop into different types of organs and tissues. And when you're developing as an embryo, you can have either 2 germ layers or you can have 3 germ layers. When you have 2, you are called a diploblast, and when you have 3, you are called a triploblast. So that's what those words are referring to, the number of germ layers you have during embryonic development. So we have the ectoderm. This is seen in our diploblasts and triploblasts. That's the one that's on the outside there. So this is what would give rise to the central nervous system eventually. And then we also have endoderm. That's one we see in both diploblasts and triploblasts. The endoderm is going to give rise to things like the digestive system and the liver and the lungs. And then in our diploblasts, you see that there's a layer in here, but this is a non-living layer. But in our triploblasts, this middle layer is called the mesoderm. So this is our third germ layer in the triploblastic organism, the mesoderm. That's what gives rise to the majority of the other organs as well as the muscles. So 2 germ layers in our diploblast, the ectoderm and endoderm, 3 in our triploblast, ectoderm, endoderm, and the mesoderm. So our diploblastic organisms are going to be organisms that are radially symmetrical. So these are our jellyfish. That's our only radially symmetrical phylum. So radially symmetrical is always going to be a diploblast. And then if you are triploblastic, you are going to be a bilaterally symmetrical organism. So you can kind of correspond those together. Diploblast goes with radially symmetrical. Triploblast goes with bilaterally symmetrical. So within our triploblastic animals, so this is just our bilaterally symmetrical organisms, we also see the evolution of a body cavity. This is called a coelom. So this is a fluid-filled space that's between the digestive system and the body wall. It contains these other organs like kidneys, spleens, things like that, the circulatory system. All of that is in this fluid-filled space in the body, the coelom. So in some triploblastic organisms, they don't have a body cavity, so they are acoelomates. So if you that A in front of that word is going to be telling you that it doesn't exist here. Just like asymmetrical is no symmetry, acoelomate means that there's no body cavity. You have some animals that are classified as eucoelomates, so that EU prefix there always refers to something being a true. So this is a true body cavity. But then we have some animals that have this kind of in-between body cavities. It's called a pseudocoelomate or a false body cavity animal. So those are the 3 options for the types of body cavities that you can see in our triploblastic bilaterally symmetrical organisms. So this is what those look like. So our acoelomate here, this is going to be no body cavity. So they just have this mesoderm layer that's just filled with tissue. And it's just fully filled with tissue, no body cavity. Remember that body cavity is that space, that fluid-filled space. They don't have a fluid-filled space here, just a mesoderm layer. In our eucoelomates, so this is our true body cavity, you see that this body cavity arises within the mesoderm layer. So we've got mesoderm on the inside, mesoderm on the outside. It's fully enclosed within that mesoderm layer. Then our pseudocoelomate, it's kind of half enclosed by mesoderm and half enclosed by endoderm. So you can see that this fluid-filled cavity here, this pseudocoelom, this false cavity, the outside of it is enclosed by mesoderm, but the inside of it is enclosed by this endoderm. So it's a fluid-filled space, but it's not fully surrounded by mesoderm. So that's why this is different than our true body cavity animals. So this has evolved a few different times. We're only going to talk about it in one group today, the roundworms, but it has evolved more than once, this pseudocoelom. And then also within our triploblastic bilaterally symmetrical organisms, we see differences in this embryonic development. So let's talk about what embryonic development looks like first. So this is the early stages of embryonic development. And again, we're just talking about our triploblastic animals here. So we have our zygote, we have our single-celled structure here. They are going to undergo mitosis. So remember, mitosis, we're changing the cell number, but we're not changing the amount of genetic information. So we're just increasing in cell number here. We're going to increase to our 8-cell stage, and then we're going to keep on dividing until we get what's called this blastula. This is just a hollow ball of cells. This is what a cross-section of this blastula looks like. We've got this, this wall of cells on the outside of this ball, but the inside is hollow. So then what happens, this hollow ball of cells starts to fold in on itself. So you can kind of see how it's this outside layer starting to fold in on the inside. And this is going to form what's called the blastopore. So this pore that forms on the outside of the blastula is called the blastopore. And it will eventually form what's called the gastrula in embryonic development. And so you can see this blastopore is down here. We're starting to see these layers form, this ectoderm on the outside and this endoderm on the inside. So what I want you to know for embryonic development is just that this pore that forms on the outside of the blastula is called the blastopore. That structure is important for classifying the two different types of embryonic development that we see in our triploblastic animals. So we just need to know what this blastopore is. So our two different types of embryonic development are protostomes and deuterostomes. So this first column here is the protostomes. The second column here is the deuterostomes. We can see this blastopore right here at the top. So this is the pore that forms, and then eventually this pore that's folded in onto the inside is going to connect to the other side of the blastopore and form a tube. Right? That's our digestive tube. If you think about our bodies, we're basically just a tube, right? We've got an opening at the beginning, an opening at the end, just a tube there with the digestive tube in the middle. So this blastopore in our protostomes, this hole that forms first, is going to eventually become the mouth in these protostome animals. That hole that forms first in deuterostomes is what becomes the anus. So the mouth forms first in the protostomes. The mouth forms second in the deuterostomes. So mouth first, that's the first hole, and then the anus develops in protostomes. The anus forms first and then connects to the other side to form the mouth. Okay? So the way that the mesoderm arises is also different, but I don't want you to know that for this class. The only thing I need you to know is that the mouth is first in protostomes, the mouth forms second in the deuterostomes. The fate of that blastopore is just a little bit different. Okay, so we are going to, this is going to be our roadmap for today. I'm going to kind of keep us, guided along this roadmap here. So the first place we're going to go down to phylum Porifera, our phylum that doesn't have any true tissue. So that's where we're going to go first here. So in this phylum, these are our sponges, phylum Porifera. Again, we don't have any true tissues here, and we don't have any symmetry here. Okay? These are all aquatic animals. There are no such thing as terrestrial sponges. The majority of these aquatic animals are marine, though there are a few freshwater species. The adults are sessile, meaning that they don't move. So these animals are anchored into place, but their larvae are motile. Their larvae can swim away and colonize a new area, which makes sense, right? We don't want our larvae developing right next to where we are because we're competing for the same food. So it's beneficial for our larvae to be able to get up and move away and colonize a new place. Phylum Porifera are also filter feeders, meaning that they are going to run water through their bodies and filter out particles from the water that they feed on. So when it comes to the anatomy of sponges, so we have a large central cavity here, the spongiocele. Remember, cele is referring to body cavity here, our acoelomates, our pseudocoelomates, right? So this is a spongiocele, so just a sponge body cavity there. And then the water is going to come in through these pores on the outside called the ostia. And then it's going to go out the top, this large opening called the osculum. So we've got little pores on the outside where the water goes in. And then we've got one big hole on the top where the water is leaving. So as the water goes into these ostia, we are going to be filtering out particles that we need as a sponge. So this jelly-like substance in the body of the sponge is the mesohyl. So that's the jelly-like substance right there. And this is important because it contains what are called spicules. So spicules are what are basically the skeleton of a sponge. So they don't have bones, right? But they do have these spicules here. You can see a microscope view of those in those pictures. Those are going to give the sponge skeletal strength, allow it to stand upright and be sturdy. This is also going to contain those choanocytes. So these are what are similar to our choanoflagellates, our protist ancestors. These are our protist ancestors here, and these are the cells that line the inside of this sponge cavity. So these flagella on these cells are going to whip around, and they are going to move the water through the sponge and trap those food particles. Then those food particles are going to be brought inside of these cells and digested within the cell. So that is referred to as intracellular digestion happening within the cell. Okay, so these flagellated cells are just lining the inside of this sponge cavity. And then next to these choanocytes, we have what are called amoebocytes, and they are going to take the nutrients that the choanocyte cells got from the food particles in the water, and they're going to distribute it to the rest of the sponge. Okay, so we've got food particles going into the choanocyte cells for digestion, and then these amoebocytes are what are taking it to the rest of the body. When it comes to reproduction in this phylum, there's asexual reproduction. So this can happen by fragmentation, which is basically a piece of a sponge breaks off, and then that sponge is now a new sponge. Okay, so it's genetically identical to the parent because it's a piece of the parent that just broke off and became a new individual. But we can also have something called budding. This is like an intentional outgrowth from a parent that detaches. So it's very similar in the sense that it's just a piece of the parent that is now a new sponge. So asexual reproduction, genetically identical to the parent. Sponges can also reproduce sexually. So sponges are monoecious, meaning they're hermaphroditic. So this is when an organism has both the male and female gametes, so sperm and eggs. So ideally, we never want to self-fertilize. We want to fertilize other individuals. So the sperm of a sponge are carried by water, and then it's a bit of a crapshoot, right? We don't exactly know where these sperm are going to land. So lots and lots of sperm are put out into the environment, and then hopefully they can fertilize the eggs of other individuals. So we do have some sexual reproduction happening in these organisms as well. So this is a summary of phylum Porifera. So as far as the organization goes, we don't have tissues here. We just have some cellular organization, and we are asymmetrical. And then we're basically missing all of these other features that other animals have. So very, very simple animals here. So from this point on, all of our animals are in this eumetazoa clade, meaning that all of the animals from this point on are going to have true tissues. Phylum Porifera, those sponges are the only ones that we're talking about here that don't have true tissues. Everything else is going to have true tissues. So we've picked up true tissues on our roadmap here, and now we're going to exit into our radially symmetrical and diploblastic organisms. So this is just one phyla that we're talking about, phyla Cnidaria. So within phylum Cnidaria, these are our jellyfish, our corals, our anemones, our radially symmetrical and diploblastic organisms. So they have this radial symmetry. We can put planes of symmetry this way or that way. We have lots of different ways to put planes of symmetry in these organisms. And during embryonic development, they just have those two layers that arise, that endoderm and that ectoderm. Phylum Cnidaria also have this really unique trait. They have stinging cells. These cells are called cnidocytes. So cnidocytes, it looks very similar to phylum Cnidaria there with the spelling. So these cnidocytes contain nematocysts, which are the actual stinging structure. So within the cell, they have this nematocyst that's stored with a little barb, and then they can fire that barb out. So these stinging cells are unique to this group. No other group has cells that look like this. So phylum Cnidaria has cnidocytes that are unique to this group. They also have extracellular digestion. So with phylum Porifera, we had intracellular digestion, so digestion within the cell. So now we're having extracellular digestion, meaning that we have this cavity that we secrete enzymes into, and then the cells that line this cavity absorb the nutrients. So rather than bringing the food particles into the cell to digest them with enzymes inside the cell, we're doing it in a cavity, right? That's what happens in our stomach, right? Our stomach, we're not bringing food particles into cells. Our stomach cells are secreting enzymes into that cavity to break down those food particles, and then our stomach cells, we absorb them in our intestine cells, right? So food brought into a gastrovascular cavity, extracellular digestion. This is still internal, right? We're still doing it in our body. We're ingesting, then digesting. We're just doing it in a cavity rather than within individual cells. But in phylum Cnidaria, we have incomplete digestion, meaning that there's just one hole that food goes in and waste comes out of. So this is a two-way street happening here with digestion, food coming in and waste going out of the same hole. Okay, so it's incomplete. We don't have the one-way street yet here in animals. Phylum Cnidaria has two main body plans, and the type of body plan that you have for the majority of life kind of helps classify you into different types of cnidarians. So we have this medusa structure here. This is a very common like jellyfish type structure, right? We have these tentacles down here at the bottom with the mouth at the bottom. This really gelatinous layer helps us be really light so we can float around in the water. But there's also a polyp structure. So these are sessile, these don't move around, and it's kind of an upside down version of this medusa, right? We're anchored down here at the bottom, but then we have our tentacles and our mouth facing up at the top. So the medusa faces down at the bottom, but the polyp faces up at the top. The medusa can move around, the polyp is stationary. So this is just some of the diversity that we see in phylum Cnidaria. So we've got our jellyfish, sea anemones are in this phyla as well. This structure here is called a hydra. So this is a type of cnidarian also. Corals are in phylum Cnidaria. So you can see this zoomed in photo of a coral. You can see these little polyps that are all anchored here. So these are going to be the polyp stage. It's dominant in these corals. These are box jellyfish, so very similar to our regular jellyfish, just in a little bit different of a group. They've got more of a square shape here. And then a Portuguese man of war is also in phylum Cnidaria. This is not a jellyfish. These are actually really interesting organisms that are colonial. So there are multiple animals that have come together to kind of do different functions, either be this kind of sail or work in digestion. So it's actually a bunch of hydra that are here together working as one organism. So here's our summary of phylum Cnidaria. So now we have this tissue level organization. We have this radial symmetry, but we're still kind of missing a lot of the other things that we see in animals. Our digestion is incomplete. We don't have a body cavity. We're not segmented. A lot of those things are going to be in our later animal phyla. So we're still missing a lot of those things here in this phylum. All right, so now from this point on, not only do all of our animals have true tissues, all of our animals are going to have bilateral symmetry, and all of our animals are going to be triploblastic. So they're going to have those 3 germ layers that develop during embryonic development. Okay, so remember that within our bilaterally symmetrical triploblastic organisms, we have protostomes and deuterostomes, these 2 different types of embryonic development. We're going to talk about our protostomes first. Okay, so within our protostomes, we've got these 2 different groups. We have one group that's called the Lophotrochozoa group, and this is named because they have one of 2 structures. They either have this specific type of feeding structure called a lophophore, or they have this specific type of larva called a trochophore. So scientists kind of just mashed these words together, both lophophore and this trochophore, and now we have Lophotrochozoa is our group here. You either have one of those structures. The other group of protostomes is called the Ecdysozoa group. So this is an animal that does what's called ecdysis, which is where you molt an external cuticle to get larger. So in order to get larger, you have to molt this covering you have on the outside. Okay, so that's a process called ecdysis. So within the Lophotrochozoa group, we are going to talk about 3 phyla. We're going to talk about phylum Platyhelminthes, which are our flatworms, phylum Annelida, which are our segmented worms, and then phylum Mollusca, which are our mollusks. And then with the Ecdysozoa, we're going to talk about 2 phyla, the nematodes and the arthropods. Okay, so I know I just ran through those phyla really quickly, but we're going to break those down just like we did the Porifera and the Cnidaria. So on our roadmap here, we've picked up our true tissues, but now we've taken this exit for bilateral symmetry and triploblastic. So all the organisms that we're going to talk about from here on out are part of this group here. We're going to talk about our protostomes first, and we're going to talk about our Lophotrochozoa group first. Okay, so we're going to go down here on our roadmap, and then we'll come back up here to the top and talk about our Ecdysozoa group. So within our Lophotrochozoa group, remember these are animals that have either a certain type of feeding structure or a certain type of larva. We're going to talk about our Platyhelminthes first, our flatworms. So phylum Platyhelminthes, flatworms, tapeworms, and flukes. So these are going to be the types of animals we have in this group. So these are acoelomates, so they don't have a body cavity, right? They don't have that fluid-filled space. They just have that mesoderm layer that's just full of tissue. We do have some free-living flatworms, but a lot of flatworms are also parasitic. So like this fluke here or this tapeworm here, those are going to be parasitic organisms. But we do have some that are free-living. This phylum also has incomplete digestion. So this was similar to phylum Cnidaria, to our jellyfish, right? We've got one mouth down here. That is the entrance for the food particles, and it is the exit for the waste particles. So this incomplete digestion, this 2-way street of digestion there. And then this phylum doesn't have any sort of circulatory or respiratory system. It's all depending on diffusion or gas exchange. So that's why they're flat, right? If we were bigger and not as flat, it would be really hard to diffuse in all of the gases that we need, like oxygen, right? So if we're flat, it makes this gas exchange a little bit easier. And then these are also monoecious, so these are hermaphroditic, most of them are. So again, they have the eggs and the sperm, so they can use either one. So flatworms have some really, really interesting mating behaviors. Some of these marine flatworms are really interesting when it comes to how they mate. So when you do your animal lab, which is going to be next week, there's a video in there for you to watch about some of these mating behaviors. So you guys have that to look forward to. Pretty interesting stuff. So here's some of the diversity that we see in phylum Platyhelminthes. So this is a planarian here, one of our free-living flatworms. These 2 colorful ones here are some marine flatworms that we have. So we see some really brightly colored organisms in the marine flatworms. This one down here, this is a trematode or a fluke. So this is a parasitic organism. And then we have tapeworms also. This is just a drawing of a tapeworm. These are also parasitic. So we've got these hooks and these suckers that help for attachment to the intestines, and then these little segments are little reproductive units that can be released and become new tapeworms. So then these pictures here. So I just wanted to show these. So this is an image that won an award for being one of the best photos taken under a microscope. So it looks like a little face of a tapeworm, but these are not eyes. These are the suckers, and these are the little hooks that help for the attachment. And it doesn't really make sense for this animal to have eyes because it lives in the digestive cavity, right? So there's no light for it to see anything. So it doesn't need eyes. So these are just the suckers that allow it to attach to the intestines. And then this is a photo of a bear that's got a tapeworm coming out of its butt. So this is how the tapeworm gets into other organisms, right? It's attached to the intestinal wall of this bear. It has these reproductive units that are growing, and eventually this is going to break off. And then all of these little segments, there's a bunch of little segments here in this tapeworm, will split off and can be ingested by new organisms, and it can become a brand new tapeworm. So we've got our summary of phylum Platyhelminthes here. We start to have higher levels of organization. We're starting to get some organs that are developing. We're bilaterally symmetrical. We have what's called cephalization, which is just the formation of a head structure. So we've got sort of a congregation of sensory structures up at the front of the animal. So this head structure, cephalization. But we still don't have a body cavity, and we still don't have complete digestion. Okay, so we're still missing some of these features here. All right, so we're still in our protostomes. Remember, these develop the mouth first. So our blastopore is becoming the mouth here in our protostomes. In our Lophotrochozoa group, now we're going to move into our annelids. These are our segmented worms. Okay, so phylum Annelida, the segmented worms. This is, you see a big variety of types of worms here. They can be marine worms, they can live in freshwater, they can be terrestrial. So a lot of different types of habitats for these worms. They can be monoecious or dioecious, so they can have egg and sperm, or they could just have one or the other. So it kind of depends on the organism, but you can get both reproductive strategies in this phylum. And then we finally have our complete digestive system now. We have this one-way street of digestion. We have our mouth, the food goes into the mouth, and then the food comes out a different hole, okay? We're not backtracking out through the mouth anymore. So all of these worms have segments. So you can see on this worm here, these lines down the body. So those are all the septa that are dividing up these segments called metameres. So they all have these segments. And then most of them have little hairs coming off of their bodies called chaetae. So these little hairs coming off these worms here. If you have a lot of these extensions as a segmented worm, you are called a polychaete worm. So poly meaning many, and then this is just talking about those hairs. So polychaete worms have lots of little hairs coming off the side. So something like an earthworm doesn't have a lot of these hairs. So this is an earthworm's not a polychaete worm, but a lot of marine worms have a lot of these hairs coming off and are called polychaete worms. So this is some of the diversity that you see within annelids. So this down here, this is a Pompeii worm. So this would be considered a polychaete worm, right? We have all of these hairs coming off of the body. This one over here is a bobbit worm, the one coming up out of the ground here. This also has a lot of hairs coming off the side. This is also a polychaete worm. But then leeches are also in this group, and then earthworms are also in this group. So they all have these segments. You can see these lines on their bodies that divide up these segments. This is a photo of a giant earthworm. So some of these earthworms in this species can be up to like 10 feet long. So we get some pretty huge specimens in this phylum. All right, our summary here, we've got our organ system organization, we've got our bilateral symmetry, we've got our head, our cephalization, our sensory structures are congregated in one area. we've got our body cavity now, we've got our complete digestion, and we have body segments. So we're starting to get all of these animal traits that we see. We're starting to now have all of them in this phylum now. All right, so still in our protostome exit, still in the Lophotrochozoa group here. We talked about our flatworms, we talked about our segmented worms. Now we are going to talk about our mollusks. So phylum Mollusca, these are going to be our snails, slugs, oysters, clams, octopuses. Those are all considered mollusks. This is a really, really diverse phylum. The only one it is second to are the arthropods, which we'll talk about in a little bit, things like insects. So this is the second most diverse phylum. Most of these species do live in the marine environment, but you do also see freshwater organisms and terrestrial organisms in this phylum. They are pretty much defined by the soft body that they have. All of them have this soft body, and then most of them have a calcium carbonate shell. Not all of them have a shell, but most of them do. They all have the soft body, most of them have the shell. So all of these organisms are coelomates, so eucoelomates. They have this true body cavity. Remember, that arises from within that mesoderm layer, fully surrounded by mesoderm. They also have a complete digestive system. So this one-way street, we've got it coming in the mouth and then going out the anus, right? This complete system. Most of these are dioecious, so we're kind of leaving monoecious behind now in our evolution. We still do have some monoecious or hermaphroditic organisms, but most of them are going to be either male or female at this point. There is a wide variation in body plans. Even just thinking about the organisms in this group, you can think about those body plans, right? An octopus looks really different than a snail, looks really different than a clam. All of these are mollusks, but they just have widely different body plans. But even within that wide range of body plans, there are some shared characteristics that all of them have. So all of them have this muscular foot for locomotion. So this is kind of a cross section here of a mollusk, and down here we've got this muscular foot. So this is pretty obvious in things like snails, right? That's a very obvious muscular foot. But our things like clams and oysters also have a muscular foot that they can use for locomotion. A visceral mass is another shared characteristic. So this is just a mass, a tissue mass that contains the internal organs. So you can see it over here on the figure. It's got our intestines, it's got our gonads in it. So it's got all of these internal organs. And then mollusks also have a mantle, which is a flap of tissue that covers the visceral mass. So you can see the mantle is this blue line that's kind of lining this visceral mass here. And if an animal is going to have a shell, it's going to be secreted by the mantle. So you might not have a shell, but if you do, it's secreted by the mantle. So you can see that the shell, this kind of tan structure on this organism, is right outside the mantle because this mantle is secreting it. So this is some of the diversity that we see in phylum Mollusca. So this is a chitin here. This is a type of mollusks that has these really distinct plates on the outside. If you turn a chitin over, this is the underside of a chitin. So that's what the underside looks like. You can kind of see this muscular foot around the side there. We've got an abalone here. That's a type of mollusk. Slugs and snails are all types of mollusks. We have bivalves. So bivalves are any mollusk that has two shells that are hinged together. So like a clam or an oyster. This is a scallop right here. So those two shells that are hinged together, all of those are mollusks. And then this bottom row down here, these are some of our most intricate mollusks, the cephalopods. So this is a cuttlefish over here, a nautilus, and then an octopus and a squid. So all of these are our cephalopods. In your lab, when you do the animal lab, there's some really, really, really cool videos about cephalopods. So make sure that you're watching all those videos in the lab next week. So cephalopods specifically, I want to talk about them because they are pretty advanced when it comes to mollusks. So these are our squid, octopus, nautilus, and cuttlefish. They have a very complex brain, and they also have pretty elaborate eyes. So these are very, very similar to the eyes that we have as vertebrates. So in this figure here, you can see the vertebrate eye compared to the cephalopod eye. It's very similar. Okay, so this is an example of convergent evolution, this analogous structures that develop. So two separate evolutionary events, one evolved cephalopod eyes, one evolved vertebrate eyes, but they have converged on similar structures because of similar evolutionary pressures, right? It was very beneficial to have these complex eyes, so that's why they evolved twice. Okay, so cephalopods also have these structures called chromatophores, which are these color and texture changing cells on the outside of their bodies that allows them to change their color for a variety of purposes. It might be I'm changing my color and my texture to blend in so nobody can see me. Maybe I'm changing my color and my texture as some sort of display for females. Whatever the reason may be, they have this really crazy ability to change the color of their body and the texture of their body using these chromatophores. So again, definitely check out the videos that are in the lab next week. So here's our summary of mollusks. So we've got our organs, we've got our bilateral symmetry. Cephalization is usually present, right? We usually have this congregation of sensory structures up at the front. But things like our bivalves, our clams, our oysters, our scallops, there's no real cephalization there. But we do have a true body cavity. We do have this complete digestion, but we don't really have any true body segmentation like we saw in those segmented worms. Okay, so we have finished talking about our Lophotrochozoa group. So now we are still in our protostome. So we're still in that embryonic development where the blastopore becomes the mouth. The mouth develops first. And now we are going to talk about our other group, the Ecdysozoa group. So this is our group that undergoes ecdysis or molting of the outer cuticle. We've only got two phylum that do this ecdysis, this molting of the cuticle. We've got nematodes or roundworms, and then our arthropods. So we're going to talk about nematodes first. So phylum Nematoda, our roundworms, these are unsegmented worms. We've talked about a lot of worms today, right? But we want to make sure we keep them all straight. We've got our flatworms, we've got our segmented worms, and we've got our roundworms, okay? So we are talking about unsegmented, not flat worms, our roundworms. So this is the only group that we're talking about that are pseudocoelomates. So they don't have a true body cavity. It's not completely lined by that mesoderm. It's kind of sandwiched between the mesoderm and the endoderm, this false body cavity. We do see some free-living nematodes that live in the soil, but most of these are going to be parasitic. Most of them are going to be kind of just living within the bodies of other animals. So wide range of reproductive strategies here. They could be monoecious, so they could be hermaphroditic and have eggs and sperm. They could be dioecious and just have egg or sperm. But we also do see some asexual reproduction in the nematodes. This is primarily just when a female creates an identical copy of herself. So we've got asexual reproduction that can happen as well. We still have our complete digestive system, but now we have an exoskeleton that's made of chitin. So we talked about chitin when we talked about the fungi. So this is the same structure. We have an exoskeleton, so it's on the outside of the body made of chitin. So that's what's going to have to be molted and shed in order for this nematode to grow, right? That's the process of ecdysis. This exoskeleton is shed and a new one is formed. So this is some of the diversity that we see in nematodes. So we have things like hookworms. So this is an example here. This is a photo of heartworms and a dog heart. So those are all nematodes. Elephantitis here is caused by a parasitic nematode. This here is the Loa loa, which is the African eyeworm. It's found in other parts of the body. It's just really obvious to see when it's in the eye of someone that's infected. So we do see a lot of parasitic species in this group. Our summary here for phylum Nematoda, we have our organs, we have our bilateral symmetry, we have our cephalization. Our body cavity, our coelom, is one of those false body cavities, our pseudocoelom, but we still have our complete digestive tract. Our segmentation isn't here, but we are molting this external exoskeleton, right? We are undergoing ecdysis in order to get bigger. That's why we are in this ecdysozoa group. All right, so ecdysozoa, in addition to nematodes, has this other group called arthropods. So we're going to talk about arthropods now. So this is the most diverse phylum that we see. So we've got insects here, crustaceans, arachnids. There's a huge variety of species in this phylum. All of these are coelomates, so they have this true body cavity, and they also have an exoskeleton made of chitin, right? Just like our nematodes have this exoskeleton made of chitin that they have to shed in order to get bigger, arthropods also have an exoskeleton made of chitin that they have to shed in order to get bigger. We're going to see a segmented body here. So sort of dividing up the body into different parts. And then we're also going to see the addition of jointed appendages. So if you think about like this crayfish here, they have these jointed appendages. So this is something new that we haven't seen in our other phyla yet. Phylum Arthropoda has an open circulatory system. So while they are pretty advanced in their body plan, they don't have a closed circulatory system like we do. So they have an open circulatory system, meaning that instead of the blood being contained in vessels, it just kind of sloshes around within the body to bathe the organ that way. And they're getting their oxygen through either spiracles or gills. So this is an example of the gills of this crayfish here. That's what all of these really filamentous structures are. But then terrestrial organisms don't have gills. They're going to have spiracles. So you can see that in this top figure here on this caterpillar, these little holes on the outside of their body that air comes into. And then once it's in there, these spiracles kind of branch off into smaller and smaller tubes that kind of carry the oxygen through the body. But spiracles and gills are going to be how these animals get oxygen into their bodies to bathe these organs in oxygenated blood. So a huge, huge, huge amount of diversity in this phylum. We could do a whole semester on diversity of arthropods. But this is just kind of a snapshot of the diversity. So we've got things like spiders, ticks, and scorpions. So our arachnids here, things that have 8 legs and these really specialized structures up at the front near their mouths. We've got horseshoe crabs, things like millipedes and centipedes are also in this group. All of our insects are in this group. So grasshoppers, bumblebees, flies, ladybugs, roaches, all of these insects are in this group also. And then our crustaceans are in this group. So crabs, lobsters. This is a plankton crustacean, a copepod, and then a mantis shrimp over here. So all of these are all going to be in this arthropod phylum. And so a huge amount of diversity in this phylum. All right, and we've got our summary here. So we've got our organs, we've got our bilateral symmetry, our head region, our body cavity, all of those things are present, our complete digestive tract, and our segmented body. And the other things we have this exoskeleton that we molt, right? That's why we're in this ecdysozoa group with the nematodes. And then we also have our jointed appendages. So we have our exoskeleton that we molt. All right, so we have talked about the protostomes. We are leaving protostomes behind now, and now we are going to be moving into our deuterostome group. So remember our protostomes, that hollow ball of cells, that blastula, that pore that formed on that hollow ball of cells, that blastopore, became the mouth in the protostomes. Now we're moving into deuterostomes. That pore that forms in that hollow ball of cells is going to become the anus. So the mouth is forming second in the deuterostomes here. So on our roadmap, this is where we are. We're still in our bilaterally symmetrical triploblastic organisms, but we've taken this detour down here to deuterostomes. there are two phyla we're going to talk about, the Echinodermata and the Chordata. So phylum Echinodermata literally means spiny skin. So these are things like sea stars, sand dollars, sea urchins, sea cucumbers. All of those are in this phylum. This is a strictly marine phylum. There are no terrestrial or freshwater species. And one of the interesting things about this phylum is that they can regenerate lost body parts. So if somehow maybe this leg of this sea star here managed to get cut off, it could grow a new one. Okay, so this really awesome adaptation they have to be able to regenerate parts that are lost. So when you look at a sea star, you might be a little confused as to why we're classifying it as a bilaterally symmetrical organism, but that's because the larvae are bilateral. So this is what the larva looks like here. So this is very much bilaterally symmetrical, right? We could put a plane of symmetry down the middle of this figure here, and it has a right side and a left side. But then our adults develop what's called secondary radial symmetry, so a secondary type of symmetry in this organism. So the adults do have radial symmetry. We're classifying these as bilaterally symmetrical because of that larva. Okay, so don't get confused here. These are still considered to be bilaterally symmetrical organisms, even though the adults look a little bit different and are radially symmetrical. Phylum Echinodermata has this unique feature called a water vascular system. So this is something that is not seen in any other group. It's only seen in this phylum. And it's a way that these organisms get water into their bodies and move it around their bodies that's useful for gas exchange, nutrient circulation, and for actually moving around for locomotion. So this is kind of a cross section here of this water vascular system. So water enters through this little pore that's on the outside of the body called the madreporite. So water is entering through there. From there, it goes down into this circular structure. So we're looking at the gray structure here, the circular structure called the ring canal. And then once the water's in this ring structure, it can go out into the arms of the animal through the radial canals. And then it can move the water into these little structures here called tube feet and allow it for movement. So if you've ever seen the underside of a sea star and kind of watched it walk around, all of those little structures that are helping it move are called tube feet, and they're able to move because water is being moved into those structures. Okay, so this water vascular system, really unique to this phylum, not seen in any other group. Here's some of the diversity that we see within phylum Echinodermata. So again, our sea stars, our starfish are in this group, but we also have things like sea urchins and sea cucumbers. Brittle stars kind of look like sea stars, but they're just in a different subsection of this phylum than our sea stars. A feather star here, so a pretty unique type of echinoderm in that photo. And then things like sand dollars are also in this phylum. So strictly marine, and then we see a lot of this secondary radial symmetry. Here's our summary. So we've got our bilateral larvae and our radially symmetrical adults. We don't have any cephalization here, so we don't really have this congregation of sensory structures up at the front on the head region, but we do have our true body cavity. We have a complete digestive tract, so we've got some of those features here. All right, so we are moving into our final phylum. I know this is a ton of information, and then phylum Chordata is a big phylum, so we still have a little bit of information to go here, but just hang in with me. This is our last phylum. So we are still in our deuterostome group, phylum Chordata. So phylum Chordata are the chordates. So a lot of times when people think of the chordates, they just think of vertebrates, but there are actually a few non-vertebrate chordates. So it's not just the vertebrates here. We've got a few other species. And these chordates are defined by 4 features, and these features are present at some point in development. For most chordates, these features are present during the embryonic stage, and then they're kind of lost in the adult form. but we do have one of our non-vertebrate chordates that maintains all of these characteristics into adulthood. But there's these 4 features, and this is how we have defined this phylum, how we have grouped all of these animals together. So the first feature is the notochord. So down here, this figure, this is just an example of a chordate that has all 4 of these features that we're going to talk about. So the notochord, that's this kind of gray line running along the body of this chordate. It is a flexible rod that provides skeletal support. So if you are a vertebrate chordate, this is what's going to become your vertebral column. But if you're not a vertebrate, you just have your notochord. There's also a dorsal hollow nerve cord. So you see that it's above this notochord. It is dorsal to the notochord, right? Remember, dorsal refers to the back of an animal. Ventral is referring to the stomach area of an animal. So it's dorsal to the notochord. This is something that is derived from the ectoderm. Remember that germ layer during embryonic development. That is what is giving rise to this dorsal hollow nerve cord. And this is what's going to develop into this brain and the spinal cord. So this dorsal hollow nerve cord will eventually give rise to the brain and the spinal cord in most chordates. Pharyngeal slits, that's the 3rd chordate trait that we have. In fish, these are going to become the gills, but in terrestrial animals, these eventually end up becoming parts of the ears and I think the tonsils. Pharyngeal slits there, that's our 3rd trait of chordates. And then the final trait of chordates that we have, this postanal tail. So this is primarily used for locomotion, but in some species it can be absent or even or just reduced. But at some point there will be a postanal tail that eventually is reduced or just completely disappears. So these 4 traits, this is how this phylum is defined. It's not defined by being a vertebrate. It's defined by these 4 traits here: the notochord, the dorsal hollow nerve cord, the pharyngeal slits, and the postanal tail. That's how you are a chordate, is you have to have those 4 things at some point in your development. Okay, so we are going to kind of follow this roadmap here, this new roadmap for chordate evolution. So this is very similar to the roadmap we were following before that has all of our animal phyla. So within chordates, this is the phylogenetic tree that we have. So we kind of have this ancestral chordate, and then as we progress in evolution, we gain more and more traits that eventually lead us to mammals. Okay? So we're going to kind of follow this roadmap now here within our chordate evolution and look at all of the features that we gain as we evolve as chordates. These are some of the key vertebrate adaptations that we're going to be talking about. So we've got vertebrae that we add in. Remember that not all chordates are vertebrates. So once we do have the vertebrae, it allows us to expand our range of motion. We have the evolution of jaws, allowing us to increase what we're eating. We have the evolution of lungs, allowing us to breathe air. We have the evolution of limbs. So this is, with lungs allowing us to go onto land and colonize these terrestrial areas. And then we have the adaptation of the amniotic egg, which is a specific type of egg that allows us to reproduce away from the water. So we're slowly gaining these adaptations that allow us to move away from water and into terrestrial environments, colonized terrestrial areas. So these are the vertebrate adaptations that we're going to see as we move through our roadmap. So with our chordates that are invertebrates, we have 2 types of animals. We have tunicates and lancelets. So these photos here, these first 2 photos are photos of tunicates. These are also commonly called sea squirts because they can kind of squirt water out of their bodies. So tunicates right here. And then lancelets, these are photos of lancelets over here. Kind of looks like a little fish. It kind of buries itself in the sediment like you see here in this last photo. These are our most primitive chordates, right? These are invertebrate chordates. So they don't have most of the advanced traits that we see in chordates. So obviously we don't have the 4 chordate traits retained into adulthood here. These look very different than that photo I showed you with all 4 of those chordate traits. But these lancelets, these actually retain all 4 of those chordate traits into adulthood. So you can look at an adult lancelet and see the notochord, the dorsal hollow nerve cord, this postanal tail, the pharyngeal slits. You can see all of those in these lancelets. In tunicates, we have them as larvae, but then when we become adults, we are sessile. We can't move around. Our larvae can move around, but we cannot as adults. So that's a similar strategy to what we saw with the sponges, right? Our sponge adults are sessile. They can't move, but the larva can move. The larva can get up and move to a new place, colonize a new habitat, right? Makes sense for competition. You don't want to be right next to mom and dad if you're competing for the same resources. So once we move past our invertebrate chordates here and we get past those, we see the evolution of a cranium. So this is what's going to surround and protect the brain. So we see the evolution of the cranium, and when we see that, we get this new group called hagfishes. So we don't quite have our vertebrae yet. Hagfish don't have a vertebral column, but they do have a cranium, which our tunicates and lancelets did not. So our hagfish is our new group that's developed. So hagfish are all marine, and they are all scavengers. So this is a hagfish here. This is second photo shows their mouth. So they are all scavengers that live in marine environments. So they retain this notochord, right? Remember we said the notochord is what is going to become the vertebral column. They don't have vertebrae, so they just have this notochord as an adult, but they have that cranium to help protect the brain. Hagfish also have this really unique adaptation to produce slime as a defense mechanism. So if you're holding a hagfish, they just get really, really slimy, and then it's hard to hold them, and then they can slither away and not be eaten, right? So this photo here, this is from a car accident that happened off the Oregon coast. Must have been in 2016, 2017. Basically, there was a semi-truck full of hagfish. They were eventually going to be shipped over to Asia, where hagfish are considered a delicacy. But the truck crashed, and so all of these hagfish, were in a stressed state and started producing all of this slime, and it covered all of the cars that were involved in this crash. So this is a car covered in hagfish slime, and you can kind of see some of the hagfish down here as well. But there's some good photos in different articles about this accident. So this is just an example of hagfish slime here. All right, so moving on here, we have our cranium, and now we are going to see the evolution of vertebrae. This is going to protect our spinal cord. So once we get this vertebrae, our new group here, so we've got a cranium, now we also have vertebrae. Our new group are the lampreys. So lampreys have vertebrae and a cranium. Lampreys are marine and freshwater. Most of them are parasitic. So these are lampreys here. So you can see its eyeball is right there. These are its little gill slits there. This sucker structure is the mouth. So it's toothed and funnel shaped, and allows it to attach to other fish and feed that way. So this is an example of a fish that did have some lampreys attached to them. So this is how they are parasitic. They can just kind of suck the nutrients out of other organisms with this toothed mouth. And then lampreys also have what we call external fertilization. So external fertilization is where a female is going to lay eggs outside of her body. A male is going to come by and fertilize those eggs outside of the body. Okay, so this is something that can only happen in an aquatic environment. So as we progress in evolution, we are going to see sort of this move towards internal fertilization. But external fertilization, we have eggs outside the body, sperm outside the body. Fertilization happens externally. Okay, so moving on past our lampreys, we now not only have a cranium and vertebrae, but we see the evolution of jaws. Okay, so our hagfish and our lampreys are often classified as the jawless fishes. That's a group called Agnatha. So A, again, meaning without, and then Natha referring to jaws. So chordates with jaws. Everything else from here on out is referred to as a gnathostome, meaning an animal that has a jaw. So if you are without a jaw, you are in the group Agnatha. So our lampreys and hagfishes are jawless fishes. Now we have the evolution of jaws, allowing us to eat a much wider variety of prey. So we are now in this group called the gnathostomes. So the first group that we're going to talk about here that has the cranium, the vertebrae, and the jaws are going to be our cartilaginous fishes. Our cartilaginous fishes are our sharks, our skates, and our rays. So they have a cartilaginous skeleton, right? They don't have an ossified internal skeleton, a bony skeleton. Their skeleton is made of cartilage. They have these really interesting type of scales. So this photo down here on the left is the type of scales they have. They're called placoid scales or dermal denticles. It's very, very similar to teeth. Like their bodies are basically just covered in these little teeth. They also have a really, really large and oily liver. So that's to help with buoyancy, right? It doesn't help a ton, right? They still, if they're not doing anything to swim, they are going to be laying on the bottom of the ocean. But this liver does help them be buoyant when they're swimming around. It makes it a lot easier to adjust their place in the water column if they have some assistance with buoyancy. So cartilaginous fishes are really interesting because they have internal fertilization, which in the rest of the fish we don't see. So this down here, this is an example of two shark species. These right here, these long structures on the inside of those fins are called claspers. Those are only seen on the male sharks, and those are to help with this internal fertilization for sperm transfer. So this is our female. She does not have those claspers. So these structures here help with that internal fertilization. But even though we have internal fertilization, we still have some that are laying eggs, but then some that are giving live birth. So a weird range of reproductive strategies, even though they're all fertilized internally. So here's just an example of a live birth for a shark here. So you can see the little baby coming out right there. And this is an example of an egg that might be laid by one of these cartilaginous fishes. These are often referred to as mermaid purses. So in our some of our skates and our rays, you see these. So the developing offspring is inside this structure there. And then within our cartilaginous fishes, we have this closed circulatory system, but we have a heart that only has 2 chambers. So if you think about our heart, we have 4 chambers. We have 2 atria and 2 ventricles, but not all vertebrates have that 4-chambered heart. We kind of need to evolve towards that efficient 4-chambered heart. So fish just have 2 chambers. They just have 1 atria and 1 ventricle. So a 2-chambered heart in these cartilaginous fishes. Here's some of our diversity. So we've got our sharks, our skates, and our rays. So we have sharks like this tiger shark here. That's what most people think of when they think of sharks. But some of our biggest sharks are actually these filter feeders. So we've got a whale shark over here. This is a basking shark over here. So these are the two of the biggest sharks. This is the biggest shark species that's out there. But these are actually don't have those big scary teeth that we think of when we think of sharks. These are actually filter feeders. And then we've got some rays down here. This is a blue spotted ray, and then a manta ray here, a very large species of ray. And then this is a skate. So looks very similar to the rays that we have, but there are some differences there. So sharks, skates, and rays are going to be in our cartilaginous fish group. All right, so we've got our cranium, we've got our vertebrae, we've got our jaws, and now we are going to be getting lungs asterisk. Okay, so this is lungs and lung derivatives, basically. So it's up here, lungs and lung derivatives, right? Because when we think about fish don't have lungs, but they do have structures that are kind of pre-lung structures. So this is what we're getting with this new group. We're going to be talking about our bony fishes now. So we talked about our fish that had cartilaginous skeletons, and now it's fish that have a bony skeleton, ossified internal skeleton. So the bony fish are the most diverse group of vertebrates. Okay, so just talking about vertebrates here, this is our most diverse group. They have this internal skeleton that's bony, right? It's no longer cartilaginous, it's ossified. The scales on the outside of our bony fish look a little bit different than our cartilaginous fish. There are two different types of scales they might have, but they're both different compared to those teeth-like scales on the outside of cartilaginous fish. Instead of having a really large liver to help with buoyancy, bony fish are going to have a swim bladder for buoyancy, which is basically a little balloon filled with air inside their body that helps with buoyancy. This is our lung derivative. Okay, our swim bladder is the lung derivative that we're talking about here. So it's a structure in their body full of air. And they have different ways that they can fill it with air. Some just do gas exchange within the body. Some can actually gulp in air to fill up their swim bladder. So this is the lung derivative that we have in these fish. In bony fish, we have external fertilization. So that's different than our cartilaginous fish. So just like our lampreys, we have our female lays the eggs, a male comes by, fertilizes them. It all happens outside of the body. But we still have our two-chambered heart. We have our one atria, our one ventricle within this group. We have our two-chambered heart still. So the bony fish are split into two different groups, and it's important that we understand these two groups because the second group is sort of a evolutionary link between living in the water and living on land. So the first group that we have are the ray-finned fishes. So this is kind of just a sketch here of these ray fins. So bony rays that are stiff. There's no muscles in these fins. You can see right here, there's no muscles on that fin. It's just kind of this ray projection there. So this is where we see the huge variety that goes into this group, into our bony fish group, are all from our ray-finned fishes. So just some photos here. It's a paddlefish, a seahorse, a salmon, some fish that we see in coral reefs. And then this one down here is a mola mola, or the ocean sunfish. This is the largest bony fish, most dense fish that you see. Super weird fish right there. That's the mola mola. So bony rays that are stiff with no muscles. The second group is the lobe-finned fishes. So this is the evolutionary link to terrestrial animals. So over here, this is our ray fin from the previous slide. This is now our lobed fin. So you can see how we have these bony rods into the fin. Okay, so we've got this bony projection into the fin. There are muscles in this fin now. So kind of getting that limb-like structure. Okay, so there are just two types of fish in this group. We've got the lungfish, which can breathe air and come up on land. And then we've got the coelacanth here. The coelacanth is super interesting. It was thought to be extinct for a really, really long time, but then was discovered at a fish market in, I think, Indonesia. And it was kind of amazing because they thought it had been extinct for millions of years, basically. But these coelacanths have these bony projections into their fins, and they can kind of, on the bottom, like on the ocean floor, can kind of move those fins in a way that's very similar to walking along the bottom of the ocean. Okay, so this is our evolutionary link to terrestrial animals. All right, we've got our cranium, our vertebrae, our jaws, our lungs, and lung derivatives. And now we are going into what are called the tetrapods. So all of these animals from here on out have legs. They have 4 limbs on their body. Tetrapods just referring to 4 feet, basically. So we've got these 4 limbs that are allowing us to be on land. Okay, so the first group we get here are the amphibians. So our amphibians are terrestrial, but they are highly, highly dependent on water. Right? They need water for this reproduction. We still have external fertilization. So eggs are laid by the female, fertilized by the male outside of the body. And then we also have this really amazing metamorphosis that happens in amphibians, kind of also highlighting our dependence on water still. So we have these eggs that develop into aquatic larvae. These larvae are herbivorous. They have gills, right? They are fully aquatic. But then they go through this metamorphosis, this development, where they become carnivorous and terrestrial and have lungs. Okay, so we're still dependent on water because our larvae have to have water, but our adults are now terrestrial. And then amphibians also have this unique adaptation called cutaneous respiration. Anytime you see cutaneous, it's referring to the skin. So this is where they're able to breathe through the skin. So they have lungs, but they're also able to use their skin for gas exchange. You have to be wet. Your skin has to be wet in order for this gas exchange to happen. Just again, highlighting that dependence on water. But in addition to breathing through those lungs, we can breathe through our skin. And in our fish, we had our two-chambered heart. Now, as we've moved on to land, our heart is becoming a little bit more efficient. We now have 3 chambers in our heart. We have 2 atria and 1 ventricle. So the whole point of developing these chambers is to eventually have complete separation of blood that has oxygen and blood that does not have oxygen. So in our fish, we had a lot of mixing of that. But then now we're starting to get a little bit of separation between blood with oxygen and blood without oxygen. So an extra chamber now in our amphibians, this 3-chambered heart. So here's some of the diversity that we see in amphibians, things like frogs, salamanders. So we've got a couple of frogs up here. This over here is an axolotl. So it's like a type of salamander that kind of retains some juvenile characteristics into adulthood, like these external gills here. We've got a spotted salamander down here and a giant salamander. There's just a couple of those species. And then this bottom row, I dedicated all to the caecilians because that is sort of an underappreciated group of reptiles. So they're kind of snake-like. They don't really have any limbs, and they're also blind, mostly cave-dwelling species. But they have some pretty interesting parental care strategies. So this is a female with her eggs. When these eggs hatch, what they do is they eat the outer layer of her skin to gain nutrients. So that's sort of one of their weird strategies here. But this is a type of amphibian. It's called a caecilian. All right, so within our tetrapods, we're past our amphibians now. We are getting a new group of animals called the reptiles. We have the evolution of the amniotic egg. Okay, so this specialized egg that allows us to reproduce away from the water. And these are going to be reptiles, including birds. So birds are included in our reptile group. So the amniotic egg has this hard or leathery outer coating, which is going to prevent the egg from drying out. So if you were to take a frog egg or any amphibian egg out of the water, it would desiccate, it would dry out, and that embryo would not be able to survive. But these eggs are watertight, so there's no loss of water happening. They have this hard or leathery outer layer. And then it's also surrounding a yolk that is going to nourish the embryo. So a lot of these structures in this amniotic egg are very similar to the mammalian embryos. You can kind of see some of the structures that are similar. You don't need to know these different layers. Just know that the amniotic egg prevents the embryo from drying out, and it has nourishment for the embryo all contained in that egg. So with our reptiles, we are now fully terrestrial. Being fully terrestrial allows for the expansion and colonization of lots of new areas. So there's also internal fertilization, right? Because we have an amniotic egg, we have to fertilize internally. Okay, that egg is watertight. Nothing's getting in, and the only thing that's getting out is the offspring that has developed. So we have to do fertilization internally inside the female. We also have mostly 3-chambered hearts in our reptiles. Our turtles, our lizards, our snakes, those are all 3-chambered hearts. But then we start to see some 4-chambered hearts also. So crocodilians have 4-chambered hearts. So alligators and crocodiles have 4-chambered hearts. And then all of our birds have 4-chambered hearts as well. So the most efficient type of heart to have is a 4-chambered heart. Crocodiles have that, birds have that. And then we also see the evolution of what are called thermoregulation strategies. So in the water, you're pretty much going to be the same temperature all the time. Water, we talked about this way back at the beginning of the semester. Water doesn't really fluctuate in temperature as much because it has that really high specific heat. So it takes a lot of energy to heat up water. So water is pretty constant temperature. So if you're in the water, your body is pretty much a constant temperature. But once you move outside of the water, once you are terrestrial, you need to figure out either a way to cope with a wide range of internal body temperatures, or somehow you need to figure out how to maintain a constant body temperature. So now that we're terrestrial, we have to have these thermoregulation strategies. So there are two different types of thermoregulation strategies. Two different types, and then kind of two different ways to get heat. So when we're talking about where an animal gets their body heat, you can either get your body heat from an external source like the sun. So you can be ectothermic, that body heat from an external source, or you can be endothermic and get your body heat from an internal source. So that's like us. We get our body heat from our metabolism. It is an internal source, but things like lizards or snakes, if they get cold, they need to move out into the sun to warm up their body, right? They're getting that heat from an external source. But then you can also have different strategies on how much change you can tolerate in your body. So we are considered to be homeothermic, right? We have a very narrow range of body temperatures that we can tolerate. But there are other species that are considered to be poikilothermic, meaning that they can have a really wide range of body temperatures. So this is primarily if you're going to be ectothermic, if you're getting your heat from an external source, it's really beneficial to be able to tolerate a wide range of temperatures because your external sources can vary a lot, right? The sun is up and the sun is down, right? You can't have the heat of the sun all the time. So you're going to have to be able to tolerate a wide range of body temperatures. So this is something that I really want you guys to know. There is no such thing as being warm-blooded and cold-blooded. Those terms are not real. You can be endothermic or ectothermic. You can be poikilothermic or homeothermic, but you are not warm-blooded. You are not cold-blooded. So please, please, please never use those words. On the animal quiz, if I ask a question and you answer it with warm-blooded or cold-blooded, I might actually take off points because that is not a real thing, okay? We need to use our thermoregulation words like ectothermic and endothermic, or poikilothermic and homeothermic, okay? So this chart just kind of shows the overlap between where animals are getting their heat and the amount of variation that they can tolerate. So most birds and mammals are going to be endothermic and homeothermic, okay? There are some exceptions, there's always exceptions, but most birds and mammals are going to produce their body heat internally and maintain a pretty constant body temperature. Okay? There are some birds and mammals that can be considered poikilothermic, meaning that their body temperatures vary during certain time periods. So if you are a bird or a mammal that maybe goes into some sort of hibernation or torpor, where you are kind of shutting down some of your body processes, your body temperature might drop. So we're not maintaining a constant body temperature in that instance. So we can see that happen here. But most of the time, if you're endothermic, you're also homeothermic. And then down here, most of our ectothermic animals are also going to be poikilothermic, right? If you are getting your body heat from external sources, you're going to need to tolerate a wide range of body temperatures because you never really know where your body heat's going to come from, right? Or the sun's going to set at the end of the day and you're going to have to get colder. So most fish, amphibians, and reptiles, those are mostly going to be ectothermic and poikilothermic. We do have maybe some tropical reptiles that can be close to being homeothermic, but it's really hard to be ectothermic, to get your body heat from an external source and maintain a constant body temperature. So it's a really hard thing to do. So it'd have to be a pretty unique circumstance for this box to be met here. Most of the time it's going to be this box, endothermic and homeothermic, and ectothermic and poikilothermic. Most of the time those two are going together. All right, so here's some of our reptile diversity. So we've got geckos, lizards, snakes, turtles, and tortoises are all reptiles as well. So I threw in this picture here just because I think it's really fascinating. So this is a leatherback turtle, one of our sea turtles. This is the inside of the mouth of a leatherback turtle. So these primarily feed on jellyfish. So when the jellyfish goes in, all of these projections coming down their throat prevent it from coming back out. So that's just a really cool adaptation for feeding seen in this species, this leatherback species.
We've got a tortoise here, Galapagos tortoise there, and then an alligator and a saltwater crocodile. So our crocodilians, these are the ones with our 4-chambered hearts. The rest of these all have 3-chambered hearts. So birds are considered part of the reptile group, but we're going to talk about them separately because they have some pretty unique adaptations. So birds are the direct descendants of dinosaurs. These are endothermic, so they are producing their body heat internally. The rest of our reptiles are not endothermic, they are ectothermic. Birds also have a 4-chambered heart and an extremely efficient respiratory system. You have to have an extremely efficient respiratory system in order to fly. It's very costly to fly. So they have this really unique respiratory system that separates the air that has oxygen, new air coming in, and oxygen and air that doesn't have oxygen that's going out. Right? If you think about our lungs, we breathe in air that has oxygen, and then we exhale air through the same pathway that no longer has oxygen. So we have air coming in and air going out. So there's no way for us to constantly be taking in air and getting oxygen from that air. But birds have this really unique system where they are kind of just constantly taking air in, and it's never overlapping. It's just new air going over those membranes to take that oxygen out. And then there are some other unique adaptations birds have for flight. So they have feathers. So these are reptile scales that have been modified. So reptiles have those keratinized scales on the outside of their body. They're scaly. Birds have feathers. These are just reptile scales that have been modified. Birds also don't have teeth, kind of lightens the load in the head, make it a little bit easier to fly if you're lighter. And then we also see some hollow bones in birds, not all species. So our aquatic birds like penguins, and then birds that run around a lot like ostriches and emus, those don't have hollow bones, but other birds do. So there's a photo of that here, these pneumatic bones, these hollow bones. So it is not to make them lighter to help with flight, but it's to add structural support for the flight. So that's why they have these hollow bones, this added structural support. So here's some of our diversity we see in birds. We've got our ostriches and our penguins, right? These do not have hollow bones. But then we have things like songbirds, we've got a blue jay, we've got owls, vultures, bald eagle. This is a putu here. So a wide variation we see in birds. All right, we have now covered all of the groups of chordates except for the mammals. So mammals have all of those features that we've talked about so far, and now we have a new feature also. We have hair. Nobody else has hair except for mammals. So mammals use hair for a variety of things. So they can use it for insulation. So here's an example of a sea otter down here. Sea otters have the densest hair of any mammal. It's about 1 million hairs per square inch. So insulation there on that sea otter. It can be used for camouflage. So here we've got some camouflage happening there with a big cat. Sensory structures. So here is a seal that has some pretty intense whiskers, some vibrissae here. Those are just modified hairs, right? So helping with sensory structures there. And then sometimes hair is modified into defense structures. So our porcupine here, these quills that it has are just modified hairs. So hair is a new trait for this mammal group, but it can be used for a lot of different things. Mammals also have mammary glands. That's why they're called mammals. So they have the ability to produce milk to feed their young. This milk varies a lot in composition, right? We think about like whole milk that you get from the grocery store is like 3% fat, but there are some species of mammals that have milk that's up to like 60% fat. Some of the like Arctic species have these really, really rich milk that's produced, but all mammals have these mammary glands. Mammals are all endothermic, so we're producing our body heat internally. And then all mammals have a 4-chambered heart. So just like birds, we are endothermic and have a 4-chambered heart. Okay? And then mammals also have something called heterodont dentition. So hetero just meaning different, dont referring to teeth. So most of these mammals are going to have a variety of types of teeth. If you look in your own mouth, you see heterodont dentition. We have incisors, we have canines, we have premolars and molars, right? these teeth look different. So we have this heterodont dentition. Reptiles don't have heterodont dentition. They have homodont dentition, meaning all of their teeth look the same. Okay. Some mammals have heterodont dentition. The only one I can think of off the top of my head would be things like dolphins. But most of them are going to have these teeth that serve different functions. So there are three categories of mammals. We have egg-laying mammals, which are called monotremes. We have marsupials, and then we have placental mammals. Those are the three types of mammals that we can have. The egg-laying mammals are monotremes. It's just a couple types of animals here. We've got the duck-billed platypus, and then the echidna, or the spiny anteater is what it's called. There's only one species of platypus. I think there's 3 or 4 species of echidna. So these are mammals, right? They have hair, they produce milk, but they don't have the structures internally that are needed to develop their offspring internally, right? They don't have a placenta like most mammals have to develop their young inside. So they do lay eggs, but they still secrete milk from glands and feed their offspring with this milk that they produce. So the qualifications to be a mammal are having hair and producing milk. Monotremes do those things. They don't have teeth, they don't have nipples, but they still are mammals. They still secrete this milk just through some glands on the mother's stomach. So a platypus and an echidna there, those are our monotremes, our egg-laying mammals. Our marsupials, we have a lot of different types of marsupials, kangaroos, koalas, wombats. So a lot of these marsupials are found in the Australian continent. Marsupials have an offspring that is born very early in development. So you can see it right there. This has already been born, right? It's clearly not ready to survive on its own. So it's going to complete its development outside the mother's body, but inside a pouch. Okay, so that's what's happening right here. This is a kangaroo that is inside the pouch, going to complete its offspring inside the pouch, like this little Joey here. We only have one species of marsupial in North America. It's the Virginia opossum. So this one right here, this is our only marsupial in the United States. But a lot of these marsupials we see in the Australian continent. And then the third group of mammals we have are our placental mammals. This is most mammals that we're talking about. If you are not a marsupial and you don't lay an egg as a mammal, you are a placental mammal. So this offspring is going to finish the development inside the uterus in the mother's body. So a huge variety of diversity here, right? This could be a whole semester as well. So we've got an elephant up here. This is a manatee, and this is a hyrax. I put this hyrax in here because this is actually really closely related to the elephant, which I just think is super fascinating. And these manatees are also pretty closely related to the elephant. So these are all pretty closely related groups here. We've got things like sloths and primates, right? Humans are in that group of primates, but within the primates, you see this division between monkeys and apes, right? Things that have tails, things that don't have tails. We've got armadillos, our lagomorphs, or our rabbits. These are not rodents, they are different, different kind of skeletal structure there. We've got rodents. So these are capybaras, the largest rodent species that we have. Things like carnivores, like wolves, big cats, seals. Those are all carnivores. We've got hooved animals like these bighorn sheep, giraffes, cows. All of those have hooves. Our cetaceans, like whales and dolphins, which are actually closely related to our hooved animals. We've got things like hedgehogs, and then we've got things like bats, right? This huge, huge variety of mammals that have kind of filled all of these different niches based on what they eat and how they move and where they live. So this is the last slide here. I know it's been a long lecture, but this is just kind of summarizing all of the phyla that we talked about. And I like it because it has these big groupings over here. So you can kind of see all animals, animals with true tissues, animals that are bilaterally symmetrical, and then we've got our deuterostomes and our protostomes here. So just kind of some descriptions and then some big classifications as well. Okay, so this is the only lecture for this week because it's so much information. I just want you guys to kind of parse it out over the course of this week. And then the lab next week is really going to help drive this home. The lab this week is all about plants. So that's really going to help drive home what we talked about last week. So then next week we will have our animal quiz. So make sure that you are comfortable with what the different phyla are, right? What kinds of animals are in those phyla? What makes that phyla unique? Why is this group different than all the other groups? Okay, so these distinguishing characteristics are going to be really important for you guys to know. All right, so I'll go ahead and stick around for a couple minutes in case anybody has questions. But otherwise, I will talk to you guys next week during next week's lecture.